Welcome friends. In this video, I would like to discuss about the Lac Operon in the presence of glucose or in the absence of glucose. So Lac Operon behaves in a different manner. Lac regulation of Lac Operon, regulation of Lac Operon in the presence and absence of glucose. Now in the presence and absence of glucose, the Lac Operon behaves in a different way. So in the presence of glucose, so when the in the presence of glucose, in the presence of glucose, in the presence of glucose, the Lac Operon is expressed. Lac Operon is expressed at low level. It is expressed at a low level. There is the level of, due to the presence of the glucose, the Lac Operon expressed at a low level, and the concentration of cyclic AMP is also maintained at low level. This is cyclic adenosine monophosphate, cyclic adenosine monophosphate is also maintained at a low level due to non-activation of an enzyme, that is adenylate cyclase. So the concentration of CMP is also low, and there is no activation of catalytic reactive protein and catabolic reactive protein, and no formation of CAMP and CRP complex takes place. As these are not formed, the cap site, catalytic activation protein site, remains unoccupied. It remains unoccupied, and due to this, the RNA polymerase cannot bind itself. RNA polymerase cannot bind itself to promoter of Lac Operon, to initiate gene expression. So the gene expression is lowered. It is taking place at a very low level or no level, because the concentration of CMP is maintained low. No activation of CRP, that is the catabolic reactive protein, and no formation of the CMP and CRP complex takes place. The cap site remains unoccupied, and RNA polymerase cannot bind itself to the promoter of the Lac Operon. So the RNA polymerase the cap site, which is near the Lac Operon, it remains unoccupied. Though the pressure is not there on the operator, this cap site remains unoccupied because there is no formation of CMP and CRP complexes there. It is not there, hence the RNA polymerase fails to attach itself to the promoter of the Lac Operon to start gene expression. RNA polymerase cannot bind itself to the promoter as long as this complex is not there. Now when the glucose is not present in the medium, when the glucose is not present in the medium, that is in the absence of glucose, in the absence of glucose, the Lac Operon expressed fully. It expressed fully because the concentration of cyclic CMP is more. That inactivates CRP and there is a formation of CAMP CRP complex. That this complex attaches itself to CAMP plus CRP attaches itself to the cap site, and that attachment of the CRP and CMP complex facilitates the attachment of RNA polymerase to the promoter of Lac Operon, and promoter of Lac Operon, and the gene expression starts. The gene expression of the Lac Operon, that is the synthesis of the 3 types of enzymes starts. Gene expression starts. So in the absence of glucose, the gene expression is there. Gene expression of the Lac is there. The RNA polymerase cannot attach itself when there is no CAMP and CRP complex on the cap site. This is the cap site, and these are the structural genes of the Lac Operon A, Y and Z. Though the pressure is already have detached itself due to the presence of an allolactose in the medium, the RNA polymerase cannot bind itself unless and until the cap site is, this RNA polymerase will bind itself to the promoter when the CAMP and CRP occupies the cap site. This activates the attachment of the RNA polymerase to this promoter of the Lac Operon, and the gene expression is on. Here the gene expression is off because there is no formation of CAMP and CARP. The CAMP and the CRP complex acts as an activator. It acts as an activator, and the gene regulation with the help of an activator is known as the positive regulation. The regulation is positive. The regulation is positive when there is an attachment of this activator. This is positive inducive regulation, and without the help of that, and when there is no inducer, rather it is a co-repressor, that type of regulation is known as positive repressive regulation. So the gene regulation is positive when there is an activator, and it is negative when it is occurring without the help of the activator. That is known as the negative, and this is the positive inducive and the positive repressive regulation here with the help of an activator, and the activator is CAMP and CRP complex. So when the glucose is present in the medium, the Lac Operon is expressed at a low level, and when it is present, it is expressed fully. There is no level or very low level of CMP in the medium that is not activating the CRP, and there is no formation of this complex, and the cap site remains unoccupied by this complex, and RNA polymerase fails to bind itself to the promoter, to the Lac Operon for gene expression. So the gene expression, when the glucose is present, the gene expression of the Lac Operon is off. When the glucose is not present in the medium, the Lac Operon is expressed fully, and due to the absence of glucose, there is a considerable amount of CMP is produced that activates CRP. There is a formation of CMP-CRP complex, and this complex attaches itself to the cap site, and that facilitates the attachment of the RNA polymerase to the promoter of Lac Operon. Then the Lac Operon gene expression starts. So this is all about the regulation of gene of Lac Operon in the presence and absence of glucose. Hope it is understood by you. Thank you.